Hi, I'm Orly Frank from BeaconQ. Today I will talk about our challenge, data mining of clinical documents. So this talk I will introduce a little bit about BeaconQ. I will explain why we are focusing on tables. I will talk about the sources of the clinical data, the challenge that we are facing, and the solution that we chose. A little bit about BeaconQ. BeaconQ is an Israeli startup established in 2016 by our CEO, Yoran Bar, and the co-founder and our VP product, Ilan Karmeli. The customers are actually pharma companies and CROs that are generating the clinical data documents. Our product, Verify, is actually AI-based solution for clinical data analytics. The product enables improving the data quality, de-risking the submission, and also accelerate the time to market of the drug or the vaccine that the company is developing. So actually, why we focus our solution on tables? Probably you're all aware of the COVID situation and the vaccine that is now developing in different companies. So you heard about the process of submission thanks to the FDA for regulations. So how they organize the data for this submission? Actually, the most widely way to organize the data are tables. Tables are not used only for clinical data documents. It also is the widely way to organize data for other research data analysis. Also, if you look at tables in a human eye, you can see that tables present for you the big picture, but also you can drill down to small details of the data. The sources of the data, where it comes from. So actually we are focusing on two types of data. The first type are statistical programming. The data is produced by professional people that are doing analysis and using statistical programming like SAS is the common program in the market. This program producing digital files. These files are using during the regulation submission and are submitted, for example, for the FDA. The other sources that we are using and processing are data that is created in reports. This data is created by a human in a free text, like Word, Microsoft Word. It is also a digital file and also used as part of the regulation process. But it's a more free text document. If you look at the statistical programming, it's a little bit more strict. It has more structure, and this is more a free text document. So let's think a little bit about our challenges, what we are facing when we try to analyze these documents. So the first challenge is actually the fact that clinical data tables are designed for human eye. People are looking at the tables, people are looking at the files, and it is not designed to be consumed by a program. There are many formats that created in this world. The old ones created by statistical programs used to be TXT and RPT. Now it's more common to use HTML, LTF and PDF by statistical program like SAS. It has many plugins to create tables in different formats. Also for the medical writing, the most common way is using Microsoft Word that creates docx files and also PDF files. The other part of the challenge is the fact that the data is unstructured. If you are looking at this image, at this table, you can see the table, but the table exists only in your eyes. It actually doesn't exist in the file itself. The file itself is unstructured. This is a RPT file that is only text characters. So many of the formats, also in PDF, we can see that it's unstructured. The structure itself of the table doesn't exist in the digital file. On the other hand, there are files that has tables, but the tables exist only for the layout. It's really commonly used in HTML that you have a table only to set the elements on the page and not actually data tables. So what we are doing for solving this problem. So I will present a little bit breakdown of our solution, how we are processing this data. It is a solution, this solution is actually used in our product, verified in production. So the first step that we are using is parsing the data. The data is converting to our unified representation. From each of the formats, we are converting to one unified representation that enables us to have unified algorithms that can process this data. So this is the first step. It is a little bit changed between the formats, depends on how complex the format is. The next steps that we are using is actually defining the cell structure. So here there are two examples of different tables. You can see that cells has a text and also some styling information. And this, in your eyes, can divide the cells to different types. You can see that there are header cells, like the headers that gives information about the tables. There are data cells that are just numbers. And also there are cells that are mixed together, both headers and data. For example, the big N notation many times has also data and the header around it. So we have the different cells in the table, and now we want to understand what is the meaning of the data in the table. If you are looking at these numbers, you can see that there are cases that you can see the same number, but it actually have a different meaning. So how we can understand the data in the table. So in order to understand the data, you have to take consideration all the context of the data. The first thing that you want to see is the headers that influence the data. If this is the number, you can see that there is a header above it, and above this cell there is a different header, so it can have a different meaning. There is the table title, the title above the table that can also suggest the meaning of this number. You have also in the table the footnotes. There is many times there is a lot of information written here to explain the data. Besides looking just on this specific table, there is also the context of the whole study, and of course the context of the domain that we are in the clinical data area. For example, in this world, if you are looking on the shortcut ITT, everybody would know that this is intent to treat, but in other world, it can mean really a different thing. So this is the things that we are using in order to understand the data. This table is actually an example of a table written by a medical writer. So the basic processing in our platform. This is the basic steps that we are using in order to process the table data. The first step is actually parsing. What we are looking for is converting the raw files, the PDF, HTML, docx files, into an abstract representation. We are taking into consideration both the text in the file, but also the styling. For example, if the cell is bold, if there is a bold line underneath, all these things that help us as a human to recognize information about the structure of the table. It helps us as a human recognize if it's a header or data, so we need to consume that also, not just the numbers. The next steps is actually some text processing. You can see that we want to resolve all the shortcuts in the data so we can have the full meaning of the data. We are cleaning the text, so it helps us to analyze the next steps. The next step in our solution is actually doing classification of the different cell types. We can know which cells are headers, which cells are data, and this helps us to understand the context of the data cells. When we have the classification of the cells, now we can try to understand the scope of the headers. We are building the headers hierarchy, and then we can get for each data cells all the information that influence this cell. When we get all these steps, now we can start to understand and get all the information for this specific cell. For example, is this cell is female data? Is this cell is a specific age data? All the information that is used can be used in other algorithms for this specific cell data. We are using here all the table information, the study information, table title, table footnotes, and all the hierarchy that we understand in the previous steps. So what we do with all this information, this is the basic of BeaconQ algorithms. So BeaconQ product verify is doing validation of statistical output. The statistical output by SAS program is verified automatically by different algorithms that we developed on the base of the table processing. These verification algorithms is actually using the basic information that we retrieved from the table mining, and we can identify the different groups exist in the data, the different groups, the different subgroups, and match the different data points in the data. So these algorithms can validate a single data table, understand the data points and the subgroups in the single data tables, but actually the most powerful tool is validating cross tables data and validating the whole data of the study. This enables us to reduce the clinical processing timeline and also increase the data quality, since the validation can find errors in the data. So I hope now you understand a little bit about our challenge of data mining of the clinical documents, and I will be happy to answer questions.